should go through that too.
Hi.
Hello.
Not just you. Hello. You were the one I happened to be looking at. Hi. Gosh, I haven't seen you in like a month. No. Just kidding. How'd it go? On day and Wednesday.
E-Days? It was great.
How was E-Days?
It was
Who
fantastic.
won the keg race?
Which one?
Mining did?
Oh, yeah, Department of Agriculture.
Were they standing in mud when they were done?
I did not check. Cops came and broke it up before everybody could finish. Dudes and noise complaints. Yeah.
I would just stuck there with
And public urination.
Really?
Yeah.
Did anyone get hurt?
Huh?
Did anyone get hurt?
Not that I know of. They're just kind of
We're safe at this school.
like, yeah.
Yeah.
I attended a keg race one time and I decided I would not go back. I do not want to be there when someone dies. No, I think it's dangerous. And you know I love beer, but that's not how beer is supposed to be. So anyway, I'm done being a dad. The canoe race is pretty cool. I didn't see any of you in the canoes. None of you? Sissies. Okay. How did things go with Josh? Yes? One of you was told to not speak anymore. Okay. So he talked about, sorry, before we get into class stuff here. Yeah, so his background is very similar to mine. Did he share commercial breaks with you?
One or two.
One or two? Did he call them commercial breaks?
Only afterwards.
These are called commercial breaks. Did you ask for commercial breaks?
No, not usually, no.
What were his stories?
Radiating grapefruit.
Yeah.
That was cool.
They were radiating grapefruit for what purpose?
Instead of GMOs and,
Oh, so they're just radiating it to script the genome.
yes.
And then they would naturally select.
For red, or just red.
Whatever else happened.
Right. So you guys know the story of the American chestnut trees?
Yeah.
Great. Can you tell us the story?
There was a bunch of chestnuts all across America, and then I think people ate them or something. Or there ate them or a bug, and they all died, and now there's like a few left. But
Yes.
that's all I remember.
So you know how common an oak tree is in the U.S.? American chestnuts were that common. And they are almost extinct. A fungus got over here from Asia and infected them and killed them. And about 6 survived.
I thought there was a place that was like full of them because the fungus couldn't reach it.
Yeah, there's a place in Michigan that's surrounded by water.
Yeah.
That survived. So about, I don't know, 6 to a dozen maybe survived. And they've been breeding them together. Someone has taken American chestnut seeds and put them in a box with uranium or radiation type stuff. And after a while, took them out of the box and grew them. And some of those seeds have greater resistance to the fungus. Is that genetically modified organism?
Yeah.
Would you consider that the same genetic? Some people would say genetically modified corn is a really bad thing. Would you say that type of radiation, that random genetic variation that is occurred by radiation, is the same or different from the GMO?
It's worse.
It's worse?
Yeah.
Who said that? Why is it worse?
Because the radiation, you don't know what genetic changes are happening. At least with the corn, it's controlled, you can see observable effect. And it's negative, if you don't do that, you change something else and see how that works.
Okay. What do you guys think? Anyone else? We're way off topic here, but I'm interested. I had never considered that perspective before. I think most people would say the corn is worse. You agree with that?
Yeah. So you have no idea what else is like, it may be resistant to the fungus or the grapefruit may be red, but you don't know what else you've changed in it or what else you might be affecting that may affect humans or
So it could grow fangs and sting people.
Yeah.
Okay, yeah, I hadn't considered that. I think most people would say when you do a specific directed change, in my experience, people my age would say, when you make a specific directed change, you take this gene from this thing and put it in here. First of all, you're doing Frankenstein type stuff, then you're playing God. But corn can breed with other things. And so now that's going to get into other populations and get around the place that shouldn't. That's way off topic, but that was the grapefruit. There's people have done that kind of stuff before. Let me put it this way though. I mean, what if you have a plant that grows in an area where there's naturally a lot of uranium? How is putting the seeds in a box with radioactive material different than something that grows in an area where there's a lot of radioactivity in the soil? Or on a mountaintop where it gets exposed to more gamma radiation from space than something at sea level? Maybe one is worse than the other, but one's more natural. Okay. Final exam time.
Tuesday at 3:15.
Tuesday at 3:15. In lab, I came up with an even better solution.
Wait, wasn't this the last exam time? Was it like Thursday or something?
Yeah.
Yes.
So now it's Tuesday?
Your choice.
Doesn't either work.
So it's Tuesday or Wednesday. In lab, what was the idea I came up with in lab?
Take home.
A take-home exam that's due Thursday. So that it can be done at your preferred time. I like that idea, if we could do that.
As long as it's not
So-
simple.
If anyone wants to object, send me an email. If there's even one objection, we will have it in class.
And then they're gonna die, so.
I'm just joking, no.
Double-stranded on it.
Interesting message in terms of yeah, so this weekend I write a take-home exam for you guys on the stuff we've covered up to this point. And then we will have more material until final exam time. And that material will consist of mutations and cancer, genetics of cancer.
So we're only gonna have 4 tests total?
Yes.
Okay.
So the next 2 tests are both take home?
Yes.
When's the take home?
And the final exam is not comprehensive. It is only on the material we cover up until the
When's the next one due? The next.
So if I get that to you on Monday, when is a fair time to get that back?
Wednesday.
Friday?
Friday.
Friday.
Oh, what are you gonna do with it? Like,
Senior design.
wait, what?
Okay.
Busy. Next game.
On Blackboard, there are 4 PowerPoints for this unit. I
Okay.
believe there are more, if you
Okay.
could post them, but I'll email you
Thank you.
and leave you notes.
Okay. Okay. So, Josh talked about complementation assays, yes? Do you guys understand that? Complementation assays? I have, say, in my laboratory, I am able to grow, I have a mutant coffee plant in my lab. And the coffee beans are naturally decaffeinated. They do not make caffeine. So you can make coffee and get the joy of drinking coffee without, I don't care if it's caffeine from coffee, or like the jittery stuff. Someone over in France, it's a plant that they grow that also claims to do that. If we breed them together and we get a progeny that also cannot make coffee, do we have mutations in the same gene or a different gene? Same gene. Does anyone struggle with that? Okay. So those would be null, so we've knocked out some gene in the pathway. If we selected a whole bunch of coffee plants that cannot do this and we made this chart. No, a whole bunch is half a dozen. I know that's negative, why?
That's the same thing.
Yeah, I'm breeding it to itself, of course it can. What would I refer to that in my experiment as?
Negative.
What type of control?
Negative.
It's the negative control. Good. So actually, every one of these is going to be a negative control, right? Oops. So I'm just trying to assess where you guys are on this, so I know how to move forward. Now I'm going to be confused here. All right. This row is the same as that row, so we don't even need to write that out, do we? We can just leave it like that, right? You'll know what's in there, you can fill in there, it's just down here, right? Okay, so. Is that right? I don't know if I want to draw one of these out. You guys can understand one of these charts, right? You guys, can you figure out, do you know what a complementation group is? Good. Let me finish this up then. Those have to be positive, right? Correct? Am I doing this right? I need you guys to check me. Tell me if I make an error. Did I make any errors on that? I think I did not. Anyone see anything inconsistent on that? I'm going to talk about complementation groups. So probably what I would do is I would name, as I isolated this mutant in my laboratory, I would give that, I would call it CAF1. That organism I would call CAF1. It has a mutation in caffeine, the caffeine-1 gene. Okay? So anything that I cross with this that can't make caffeine must have, by definition, or not by definition, but must have a mutation in that same gene, right? So complementation group 1 consists of what? CAF1, CAF3, CAF4, right? Got it? Complementation group 2. Which mutants are in complementation group 2? CAF2. And CAF5. Complementation group 3 has mutations in CAF6. Just CAF6, right? Does that make sense? So, does anyone not understand how I got this? Okay. So, if I breed this to number 3, the progeny from that are not able to make caffeine. So these guys, number 3, must be a mutation in the same gene. Just independently, you know, someone over in Australia did it. It just happened to be a mutation in the same gene. So they do not complement, okay? So anything where there's this negative with 1 is the same group with 1. So 2 is in the same group with 5. So the mutation I made in my lab that prevents these guys from producing caffeine. Someone in Georgia made a mutation, they did their own mutation analysis and they came up with a different mutant, although it happens to be in the same gene, right? You got it now? So the only one that 6 is a negative with is 6. So how many genes are in the pathway for producing caffeine? There's 3 genes, right? So we've got there's gene, we're going to say enzyme A, enzyme B, and enzyme C. And I don't know, this could be A, or it could be B, or it could be C, I don't know at this point, right? How might I determine? So let's see, this is xanthine. And this is some modification to xanthine. Then this doesn't have a name xanthine anymore, it's some other thing. Modified xanthine. It's like I don't remember. Aminoxanthine, or I don't remember what it was. And then there's another modification you might have with caffeine. Something like that. Okay. Can anyone think of a way that I could determine where this mutation is?
Try to grow it with modified xanthine and the other stuff.
Yeah. So I could take this plant, and to the media, or to the soil, I could add this chemical. So that every time I water, I add a little bit of this. If I do that, this guy produces caffeine. Do I have a mutation in this gene? Can't be a mutation. This gene is working. So the mutation must be in one of these. I have just complemented my mutation by providing a nutrient supplement. I've just recovered from the mutant by making the environment appropriate. Does that make sense? So I use that word complement. I've compensated for the mutation by giving it something extra. That was awesome, thank you. So, it turns out we know the pathway for the caffeine. You can go online and say biochemical biosynthesis of caffeine. You can get that and so on. And we know what these are. Okay, great. That's how you can order things like this. Any questions on this? Do you think would you call this an exhaustive analysis? What would you consider an exhaustive analysis?
Probably 50 or 100 different plant specimens.
Let's see. So you basically take seeds and you expose them to something that's going to mutate the seeds to get damage, and you breed them and you find things, or you grow those up and find ones that can't produce caffeine. So 50 or 60 would be a first pass. When they do this work, when they were doing this in the cell cycle, because Josh talked about the cell cycle, right? When they did that on a plate, they would mutate the yeast and they would streak the yeast out on a plate, and there's colonies all over the place, and they might screen 1 million colonies per plate. And it's the job of the graduate student to do stacks of these, maybe 500 plates. So they're screening through 500 million colonies and they would get 50 different mutants each time. And my expectation would be they would put together a panel of 1,000 mutants and then cross those all together. 1 to 1, 1 to 2, 1 to 3 in a very controlled experiment. Fun to be a grad student. Right? Okay. Cool. Any questions on this? Josh talked about temperature-sensitive mutations. Do you guys understand? Did he explain why something could be a temperature-sensitive? What's the cause of it being temperature-sensitive? Folding of the protein. Did he talk about that? So when we get the three-dimensional shape of a protein, we draw it on the board or see the picture. Say, oh, okay, that's the shape. You know, it's like you made it out of stainless steel and it's hanging up. Like they've got that Gordian knot thing here by the CTLM, right? That thing's not changing shape, right? Turns out proteins, the way they all fold up, they call it protein breathing. So we draw and we think of it like this, but in the real world, a water molecule is a thing that is of substance to something of this size. The water molecule, maybe it's a gnat or a bee, you know, in comparison to like my body, it might be a fly, but a fly can still get me to do things, right? If a water molecule is coming along and has some velocity and goes smashing into this, it can actually bend and distort the shape of the protein. So proteins are constantly flexing a little bit. So temperature-sensitive mutation is just one where, you know, maybe the reason this loop of this protein is hanging out close to this is there may be a positive charge here and a negative charge on this thing right here. And so they're attracted to each other. All you need is a point mutation that would change this maybe to alanine, and so there's no negative charge anymore on this loop of this protein. And now this part of this protein is no longer really interested in hanging out like this. And so it kind of flaps around like that instead. And maybe there was an attraction from here to here that was helping hold this thing together. And so now this thing flaps out like this. And now this thing is all flapping around back and forth, and the active site of this protein has been destroyed. A very simple mutation can actually make the protein breathing become so extreme that the protein falls apart. Why were temperature-sensitive mutations so important in the cell cycle analysis? You got the answer?
You can actually see where it stops in the cell cycle at the non-permissive temperature.
So you grow it at the permissive temperature, and the cell divides and grows and grows and grows. And then you just take some of those cells and you put them on your microscope slide, and then you incubate your microscope slide at the non-permissive, at the restrictive temperature, and the cells will stop, and then you get a cell that's, you know, not quite done budding. And you say, okay, well that's probably at the end of the cell cycle. So yeah, it's convenient to see. It's an easy assay. Did he talk about assays? Yeah. And he used microscopy was the assay. It's the shape of the cell, where it appears to have stopped. By microscopy, that was his assay, right? Okay. Why else were temperature-sensitive mutations important? This is really some basic stuff. Take it way, way back.
Well, it needs to be conditional so that you can actually grow it up.
Yeah.
See if it's.
So in this, I did a mutation that screws up a pathway. What if my pathway is G1, S, G2, M, like that. If I interrupt the pathway like this, if I get a mutation here so this can't go any further, great, I have a cell that can't divide. How am I getting another cell to study? Right? Does that make sense? So they had to use temperature-sensitive mediums. You make the mutation, it can't grow, great, it lives for like 12 hours and dies. Congratulations. But you can't study it anymore after that. From a very practical standpoint, you had to have a way of having the mutation affect it and then not affect it at other times. Does that make sense? Who came up with these temperature-sensitive? Was that incredibly creative or what? Who thought of that? And what happened is, yeah, I don't know who thought of it. Someone conceptually understood this is the way it happened. Someone with a very molecular focus, a very biophysical background, said, in theory, if I got a mutation that caused this to happen, this protein would be dead and I could get this cell would appear mutant in that gene, even though it's not mutant in that gene. Even though it's not a knockout in that gene. And they said, well, in theory that should be the case. Let me go see if I can actually find some things like that. And they did. Oh, incredibly bright. Whoever did that should get the Nobel Prize. We'll never get that. We'll never hear about it. Okay. Temperature sensitive mutations, complementation assays. I was talking about production of a chemical. He was talking about, did he do histidine or leucine? Histidine is a common one. Leucine is another one. So he was saying you have to provide it a nutrient. Here I'm saying you're producing some product. How does this apply to engineers, someone who's chemical engineers? You may have an interest, if you're going to be a biochemical engineer, you may someday need to work with an organism that has a pathway. We gathered up all this crude oil which got spilled into this waterway. There's all this really nasty toxins in there. We're going to throw a fungus into this bin and the fungus is going to eat that. Right? So if we have a pathway from yucky stuff, and there's probably a bunch of steps in this pathway, and the end result is going to be CO2 and water. So there have actually been studies where people have taken like really awful, nasty, hazardous, toxic stuff, had them in 55-gallon barrels, and they've just randomly taken a whole bunch of organisms and dumped them in, and they found an organism that could survive, and it ate the yucky crude oil stuff as food, and converted it to non-toxic waste. Okay, so there's a pathway from yucky to CO2 and H2O. What are the genes involved in that? How many genes in that pathway? And why do we care how many genes are in the pathway? Does anyone guess? Why do we care? We know it works, right?
Hope that it's less genes because it's less complicated, I guess.
Okay, so say it's 3 genes. Okay, awesome. It's 3 genes in the pathway. Okay, it's less complicated.
Couldn't you engineer the fungus to express those genes more so you could use less fungus?
So first I find out how many genes there are, and then I go through efforts to figure out which genes they are. He talked about cDNA libraries, right? So what you do is you find, you've got your organism that can do this. You take your organism, you expose it to mutagens, and you find a mutant that can't do this. So by definition, then you've knocked out one of these genes. You can use the cDNA library to complement, and now you've identified the sequence of the gene that helps break this thing down. And you can do that for each complementation group. Once you know the gene, then you can start playing God and exposing that organism to radiation or doing directed mutations in that gene to try to make it better. Then we know a real-world example where we took a fungus and we did this stuff. Not just yeast for complementation, for cell cycle.
I just know that there are fungus that we know do this.
Well,
And there's not, like, it's not.
yeah.
There's a lot of fungus. Oh, yeah.
In the 1920s, someone found that you can take this organism, something other than penicillin, I can't remember the name, it's escaping me right now, it's a fungus. And they went all over the United States taking samples of fungi, trying to find organisms. They'd throw up a liter of fungi in a broth media, and then they'd say, what is the concentration of penicillin that this thing produces at maximum concentration that it gets to, if I grow that in here? How much penicillin? And there's 1 unit of penicillin per liter. And once they figure out the pathway, they can start messing with the pathway, trying to optimize the pathway so the organism would make more penicillin per liter. How effective have they been? So are they at 100 units per liter yet, do you think? Did they get to 100? Yeah, definitely. About 1,000. Six orders of magnitude. You put that organism in there, and you get 10 to the 6th units per liter penicillin out.
I'm sure.
Yeah.
How do you, like, so you identify a gene and you want to express it more and you say you like manipulate the gene, like do you just go in and change like A's to, you know what I mean, like how do you
Yeah.
know what to do to actually make that work?
That's a great question. So, okay, we found the 3 genes. We know the genes. How do I make it better at doing this? Okay. So gene A, B, and C here. Okay. So if I know the gene, I can find the sequence of it in the genome of the organism. And then I can go to this and I can say, and this is literally what happens. Okay, this is 30 base pairs. I'm going to change this to a base pair number 1 to an A in the promoter. Is it now more effective? Yes, no, maybe. I'm going to change that to a T. Is that promoter now more effective that I get more messenger RNA off here? Okay, now I'm going to change it to a C.
That's just like so much time commitment.
You bet. It's 1 chapter of a PhD. And they will change every nucleotide of the promoter to every possible. And they'll say, oh, if I change that to a G, and this to an A, and then they'll say, I'm going to put them all together. I independently found what was best at each spot in the context of the wild type. When I put the optimal of all of them together, do I get an even better one? And sometimes you do, and sometimes you don't. And that'll be someone at a company is doing that to maximize the promoter to get maximum expression out there. And then they'll do things, and it comes back to the temperature sensitive stuff. We're going to get a protein off there, and that's going to fold. And here's one I actually saw. Some guy was dealing with one of the stable polymerases used in PCR. And the way it went was like, the protein had this shape where there was this loop here and there was this loop here. And what the guy wanted was a protein that was stable at higher temperatures. And he said, wow, if I measure this distance from here to here, that is the distance. If this were a cysteine and that were a cysteine, the sulfurs poking out could form a disulfide bridge. You know what I mean by that? And so this person went into the code of the gene and changed that codon so that this was a cysteine and that a cysteine. And it turns out a disulfide bridge did form, and it increased the stability of the enzyme at higher temperatures. And that was patented. It's patented. And hopefully that guy's making a ton of money off it. So you can look and say, if the active site is right here, and my chemical fits in here, and that's it. If I had something that would form a hydrogen bond here, I could get that to bind more readily into my active site. So this is what pharmaceutical companies are doing. If any of you are up for writing computer programs to do this stuff, we were talking about this yesterday. They can figure out the 3-dimensional shape of this. What they don't have is enough brain power to make the analysis that will say, what is the shape? How can I make the active site more effective for binding the chemical and processing that chemical? And we're talking $100,000 a year job minimum right there.
Couldn't you also be looking at other things not even affecting the protein structure, like some kind of inflammatory response to another protein. If you shut that off, and then suddenly this uninhibited protein is just running rampant.
Yeah, maybe there's something that binds onto this and decreases its activity.
Yeah.
Yeah, so you find a gene elsewhere which is an inhibitor of it, and you eliminate, you knock that out of the genome, right?
So there's lots of ways.
So those are directed. Those are directed ways. But the most effective way of increasing a biochemical pathway is to take your organism and grow it up in media and squirt in a little bit of ethidium bromide and grow it up, and whatever the hell mutations happen, you've got a million cells there, and you separate them out. You put one cell in each well of a big dish, and you grow them up so now they're purified colonies, cultures. And you say, is this guy more effective than the others? And the answer is probably not. Is this guy more effective? Probably not. Is this guy more? And eventually, out of the million cells you put in there, one of them is a little bit better at doing this. Where is the mutation? I have no idea, and I actually don't really care. There are ways to map it out. It's better. And now I have that thing that's better, and I'll do the directed mutations on that. And that's how they got to 10 to the 6th, mostly, is just, hey, let's shine some UV light on it. Okay, we did ethidium last week, let's do UV light this week. And next week, we're gonna infect it with a virus and see what happens. And the week after that, we're gonna take this DNA from E. coli and blast the cells with the E. coli DNA, and maybe that'll work, I don't know. And you grow it up, and when one is better than the others, like, I don't care what happened, it's better.
How do you write a paper about that?
How do you write a paper
Like explaining
and publish that?
how that would,
Yeah, you say, the organism was exposed to mutagens, and there's a whole set of mutagens that are commonly used. We exposed to ethidium bromide and did a screening, and we did exposed to UV light and did a screening, and exposed to restriction enzyme or we activated homologous recombination machinery and did a screening, and then we go, oh yeah, that's a standard way of just totally messing with the genome. It's better.
you have to explain.
Yeah, it's probably a paragraph in the paper. People go, oh, these standard techniques, got it. Cool. So, hopefully somebody will be finding ways of converting cellulose into bioethanol fuel. And hopefully at some point in my life I'll teach someone who's going to find a way to optimize the pathway of processing cellulose into bioethanol fuel, or barley into beer, or toxic nastiness into CO2, O2, or gold dissolved in seawater into gold money in my pocket, because you like me, right? Okay. This complementation assay can be used to identify an organism that's more efficient at whatever it is you're trying to accomplish. Survive on Mars. Cool. All right, I just needed to get up to speed and make sure we were all on the same page on this stuff. It's Friday. Let's go drink beer. Not too much.
Oh,